Welcome to another deep dive. This time, we're going microscopic into the world of Actinobacteria.
Microscopic, but mighty, I'd say.
Absolutely. And we're using ultimate guide here, volume 5 of the second edition of Bergey's Manual of Systematic Bacteriology.
The go-to for bacterial classification.
Basically.
Yeah.
And for all you medical microbiologists out there, this deep dive is going to highlight some pretty amazing stuff. I mean, Actinobacteria, they're everywhere, right?
Oh, yeah. Hidden heroes, you could say.
Lurking in the soil, the water.
Mm-hmm.
Even inside us. So let's start simple. What are Actinobacteria?
Well, the manual, it describes them as a phylum, a distinct phylum of bacteria.
Like their own branch on the bacterial family tree?
Precisely.
Okay. But what makes them stand out from other bacteria?
Their genes, for one thing. They've got these unique sequences in their 16S RNA genes. Think of it as their fingerprint.
Their unique ID card.
Exactly.
So you can actually see them.
Yeah.
Like, what do they look like under a microscope?
That's where it gets interesting. They come in all sorts of shapes and sizes.
Really?
Oh, yeah. Some are just little spheres, Kochi. Others, they form these branching filaments, mycelia they're called. Really complex.
Wow. So not just one size fits all, huh?
Not at all. And that diversity, it means they can do a whole lot of different things. Metabolically, I mean.
Like, they have a lot of different talents.
You could say that. And some of those talents, they're pretty important to us humans.
You mean like producing antibiotics?
Exactly. Actinobacteria. They're the masterminds behind most of the antibiotics we use today.
Lifesavers, really. And the manual, it mentions streptomyces. Like, they're the rock stars of antibiotic production.
Yeah, they're incredibly efficient, producing something like, what, 2/3 of all the clinically useful antibiotics out there?
That's amazing. But it's not just streptomyces, right?
Oh, no. There are others. Amyclotopsis, Micromonospora.
Right.
And researchers, they're finding new ones all the time, especially in these extreme environments.
Oh, like the extremophiles.
Mm-hmm. It's like, to survive those conditions, they've had to develop these incredible abilities. And those abilities, we might be able to use those to our advantage.
Like they have superpowers.
Right.
So they're making antibiotics. But what else do they do?
They play all sorts of roles in ecosystems. Decomposers, for example, breaking down all that dead stuff.
Recycling.
Exactly. And some of them, they form partnerships with plants, helping them get nutrients from the soil.
Wow. Busy little guys. So we've established Actinobacteria are diverse. They're important. Yeah. But the manual, it focuses on a specific class, right? Class Actinobacteria. What's so special about that one?
It's the biggest class in the whole phylum.
Okay.
And it's where a lot of those medically relevant species hang out.
So that's why we're focusing on it.
Mm-hmm. The ones you're most likely to encounter in your work.
Now, the manual, it mentions something called G+C content. What is that exactly?
So G+C content, it's basically the percentage of guanine and cytosine in an organism's DNA.
Two of the, building blocks of DNA?
Yeah. And a wider range of G+C content. It suggests there's more genetic diversity going on.
So it's a clue about how diverse they are.
Right. And this classification, it's not just based on, appearances.
Right.
We're talking 16S RNA gene sequencing, specific genetic signatures.
The science behind it is so cool.
It is.
Now, let's get even more specific. The manual, it talks about the actinomycetales. Why is that order important?
Because that's where we find the family Actinomycetacea.
That's where things get really interesting for us, right?
Yeah, some of the most medically important Actinobacteria live there.
So tell me more about that family.
Well, mostly they're gram-positive rods, non-modal, often forming those branching filaments we talked about.
Okay.
And a lot of them are facultative anaerobes, can survive with or without oxygen.
Flexible little guys.
Some are even strict anaerobes, though, can only live without oxygen.
Oh, interesting. And the manual mentions they often need CO2.
Right, for certain metabolic processes.
Yeah.
Like we need oxygen to breathe, they need CO2.
I see. So they're breathing CO2 instead of oxygen.
In a way, yeah.
That's pretty wild. But wait, there's a rebel in the family, right?
You mean Mobiluncus?
Yeah.
Yes. They're a bit different. Curved rods, gram variable, and get this, they're motile.
They can move.
Mm-hmm. Unlike their relatives who just stay put.
So they're the adventurers.
You could say that. Maybe that's why they're more likely to cause infections that, spread.
They get around more. That makes sense. So what are all these bacteria actually doing inside us? Or animals, I guess.
Well, it depends. Some are commensals. They just live there peacefully, part of the normal community.
Harmless roommates?
Basically. But others, they can be pathogens.
Causing infections.
Mm-hmm. especially if they get the chance.
Like if the immune system is weak.
Exactly. Or if there's some kind of disruption in the normal microbial balance.
So they're like double agents. A little scary.
It can be. That's why it's so important to understand them, medically speaking. Know which ones are friendly and which ones, well...
Which ones might turn on us.
Precisely. And speaking of those 5 genera that make up the family...
Each with their own, like, personality.
You got it. Knowing how to tell them apart is crucial for diagnosis and treatment.
Absolutely. So let's start with Actinomyces, the namesake of the whole family. What makes them special?
Well, they're known for those branching filaments, for one. And their cell walls, they can be pretty diverse.
Diverse how?
Well, they have different types of peptidoglycans. You know, the building blocks of bacterial cell walls.
Ah, so different kinds of armor.
Yeah, you could think of it that way. And to make things even trickier, Actinomyces, it's polyphyletic.
Poly what now?
Polyphyletic. It means basically they don't all share a single common ancestor. It's like, imagine a family photo album, right?
Okay.
Some of the people, they might look alike, but they're not actually related.
So it's like we've been putting them in the wrong family photo album based on looks.
Exactly. And that's where 16S rRNA gene sequencing comes in.
Ah, the DNA test.
Right. It helps us figure out who's really related.
And that's crucial for you guys, right? For accurate diagnosis and all that.
Oh, absolutely. You gotta know exactly what you're dealing with.
Makes sense. So we know they can be commensals or pathogens. What makes them flip, like, from friend to foe?
A lot of them, they're opportunistic pathogens.
Meaning?
They're just hanging out, usually harmless. But if the immune system weakens or there's some imbalance...
They seize the opportunity.
Right.
And what happens then? What kind of infections do they cause?
It really varies. Could be a localized abscess or, you know, something more serious, systemic.
The manual mentions actinomycosis. What is that exactly?
It's a chronic infection, slow growing, often involves abscesses draining sinuses.
Sounds nasty.
It can be pretty rough, painful, debilitating, and treatment, it often involves both antibiotics and surgery to drain those abscesses.
So catching it early is key.
Oh, yeah, absolutely crucial.
Which is where you guys come in, the medical microbiologists.
Exactly, and that's why resources like Bergey's manual, they're so valuable. They're like our roadmap.
To navigate that whole bacterial world. And speaking of navigating, the manual, it gives some clues about how to tell these different genera apart, right?
Oh, yeah, it's a whole bunch of things. Their shape, their metabolism, their genes.
Like we know Actinomyces, they're usually catalase negative.
Right, lacking that enzyme that breaks down hydrogen peroxide.
And Actinobacteria.
They can be either catalase positive or negative.
Tricky.
It can be. Then you've got Actinobacillum, anaerobic, need that CO2 to grow.
Right, right.
Fairbaculum, also preferentially anaerobic.
Oxygen haters. And then, of course, we have Mobiluncus, the rebels.
The movers and shakers, literally.
It's like a detective game, huh? Using all these clues to figure out who's causing the trouble.
Exactly. And the stakes are high. Got to get the treatment right.
Absolutely. Speaking of treatment, are these guys easy to treat with antibiotics?
Well, it depends. Some are more susceptible than others. Penicillin, clindamycin, those are often effective. But some can be more resistant.
So antibiotic resistance is a worry then?
It's a challenge, yeah. A growing one. Really highlights the need for new approaches, you know, and being careful with how we use antibiotics.
Right. We don't want to make things worse. But it's interesting, right? These actinobacteria, they can cause problems, but they also have this huge potential for good.
Oh, absolutely. We've talked about the antibiotics, but there's so much more out there, especially with those extremophiles.
The ones living in those crazy environments.
Deep sea vents, hot springs. I mean, to survive there, they've had to evolve some pretty unique ways of doing things.
Like special metabolic pathway.
Exactly. And those pathways, they could lead to new compounds, things we could use for, I don't know, new drugs maybe.
Like they've got all these secrets.
Right. And with antibiotic resistance being such a problem, finding new drugs, it's more important than ever.
These extremophiles, they could be like, I don't know, a gold mine. New antivirals, even anti-cancer drugs.
It's possible. It's a whole new frontier, really.
It's pretty amazing. So how do researchers even find these extremophiles?
Well, they start by collecting samples from those extreme environments.
Makes sense.
Then they use all sorts of techniques to, you know, isolate the bacteria, grow them in the lab, analyze their DNA, see what kind of metabolic tricks they've got up their sleeves. And then, of course, they test the compounds they produce, see if they have any potential.
It's like a whole different world. and it could have such a big impact on human health.
It really could. We're just scratching the surface, I think. There's so much more to learn.
So much potential. But it's not just about human health, right?
Yeah.
The manual, it also talks about the ecological roles of these bacteria.
Oh, definitely. Even the ones that aren't directly involved in medicine, they're important.
It's all connected, huh?
It is. These bacteria, they break down organic matter, they cycle nutrients in the soil.
Keeping things in balance.
Exactly. They're vital for healthy ecosystems.
Well, I've learned so much today. Actinobacteria, they're diverse, they're important, and who knows what else we'll discover about them in the future.
There's always more to explore.
That's for sure. For those of you who want to dive deeper into this fascinating world, I highly recommend checking out Volume 5 of the second edition of Bergey's Manual of Systematic Bacteriology. It's a fantastic resource.
An amazing guide.
Well, thanks for joining us on this deep dive. Until next time, keep exploring.